Right now we're going to go over the activity where we reviewed the experiments that a bunch of different scientists did, and they were able to use the results from those experiments to determine that DNA was in fact genetic material. So the first experiment we're going to go over is Griffith's experiment that was carried out in 1928. Remember that Griffith used two strains of bacteria called Streptococcus pneumoniae to infect mice. And the two different strains were virulent and non-virulent. And so the smooth strain was the virulent strain. And when we say that something is virulent, what we mean is that it can cause an infection. The other strain that Griffith used of bacteria was the rough strain. The rough strain is avirulent, and that means that it is not infectious, so it does not cause an illness. So Griffith used these two strains of bacteria, and he used them to infect mice. He discovered that when he infected mice with the rough strain of this bacteria, the mice lived. When he injected mice with the smooth strain of bacteria, which remember is virulent, then the mice died. He then tried to infect the mice with the smooth strain, which remember is virulent and would kill the mice. But if he killed the bacteria first by heating it up, he discovered that the killed bacteria did not cause the mice to die either. So in this case, the mice lived. Next, Griffith tried something interesting. He took some of the rough strain bacteria that was alive and he mixed it with the heat-killed smooth strain. So he mixed live rough bacteria with dead smooth bacteria. What he discovered is that the mice died. When the mouse died, he took the bacteria from the dead mouse and then he grew the bacteria so he could identify what kind of bacteria it was. What he discovered is that it had transformed and was actually smooth bacteria that was growing. So what happened is this process called transformation. And transformation is a change in the genotype or the genes when a cell takes up foreign DNA. So what had happened is that the DNA from the rough bacteria stayed in the rough bacteria, but there was DNA from the dead smooth bacteria that entered the rough bacteria cells, and that caused the rough bacteria cells that were alive to start producing the smooth bacteria characteristics because of the DNA that it took up. And so transformation can cause a change in one cell by taking up DNA from a different cell. Next, we're going to talk about Avery. So his experiment took place in 1944, and he used enzymes that destroy proteins or DNA, respectively, in an experiment to determine which enzyme would stop transformation from occurring. So remember, transformation is what Griffith discovered. He discovered that if DNA travels from one bacteria to another, it can cause a genetic change. Avery wanted to find out if DNA was the genetic material or if it was proteins, because they were unsure what was causing transformation, which molecule was taking that information from one cell to another. And he discovered that transformation is not stopped by a protein destroying enzyme. And he discovered that transformation is stopped by a DNA destroying enzyme. Next, we can go over how to look at the diagram. So what the scientist did is that he extracted DNA, proteins, and RNA from some bacteria. And so he got rid of the lipids and the sugars in the solution so that all he had was DNA, proteins, and RNA. Then, with some of that liquid that contains protein, RNA, and DNA, he added a proteinase, which is an enzyme that breaks apart proteins. When he did that, he grew new bacteria cells in that solution, and he discovered that S cells, which are the ones he was getting the DNA, proteins, and RNA from, appeared even though the cells in the solution he was growing, which were the R cells, did not initially have S cell characteristics. So when he broke apart proteins, S cells were still able to appear even though that culture did not start with any living S cells, which means that protein was not genetic material because it did not, when it was broken down, it didn't prevent transformation from occurring. Then he tried the same thing with RNase, which breaks down RNA, and he discovered that if RNA is broken down, S cells still appear, which means that RNA was not destroyed, or was destroyed and genetic information was still passed. So RNA is not the genetic material that's passed from parents to offspring and caused transformation. Finally, he did an experiment where he broke down DNA with DNase, which is an enzyme that breaks apart DNA. When he broke down the DNA molecule, no S cells appeared. And that's really important because if he breaks apart DNA, then the DNA can't mutate the R cells to S cells, which means that DNA is important and must be present in order for transformation to happen. So if DNA is missing, transformation cannot occur. Now we can talk about Hershey and Chase's experiment that was conducted in 1952. Hershey and Chase conducted an experiment that determined which molecule, protein or DNA, is the genetic material of the cell. Their experiment used a T2 bacteriophage to infect bacterial cells with DNA carrying radioactive atoms. So first, let's define a bacteriophage. So number one, bacteriophages are a virus that infects bacteria. So bacteriophages don't infect people, but they can infect bacterial cells. So a bacteriophage, or the T2 bacteriophage specifically, is a virus that infects bacteria. And the type of bacteriophage that Hershey and Chase used was, in fact, that T2 phage. So it's just one specific kind of virus that infects bacteria, because there's many types of viruses and many types that can infect bacteria specifically. So in this experiment, DNA and proteins were labeled with two different radioactive isotopes. So they used radioactive sulfur, which is called S35, and they used radioactive phosphorus, which is called P32. And those two different atoms are called isotopes because they have a different number of neutrons than a normal sulfur atom or a normal phosphorus atom. So we are going to identify which isotopes were used to label the following. They used the T2 phage plus radioactive sulfur to label protein. And then they used the T2 phage and radioactive phosphorus to label DNA. So then their experiment worked like this. They took a radioactive virus that had radioactive proteins in it. The radioactive proteins are found present in the coat of the virus, which is on the outside of the virus. Then they allowed the virus to infect the bacteria. So the virus injects its DNA into the cell. Then what they discovered is that there's no radioactivity inside of the cell when they're infected by a radioactive virus if it's just the proteins that are labeled. And that's because the viral coat doesn't go inside of the bacteria. They were able to discover whether or not the bacteria contained the protein coats because they put the bacteria or infected bacteria in a blender, and the blender, when they run it, takes the virus coats and rips them off the surface of the bacteria cell, and then they put that liquid that they're all in inside of a centrifuge, and a centrifuge separates things by density. And what they discovered is that the radioactivity, which was contained in the protein coats of the virus, all stayed in this liquid on the top called the supernate. And then the bacteria cells, which are more dense, all ended up at the bottom of the test tube, and that did not contain any radioactive particles. And so they discovered that the radioactive sulfur with the proteins did not enter the bacteria cells. Now we can talk about the DNA experiment. So here they used a T2 phage with a DNA, or with radioactive phosphorus. So here, again, the virus has DNA, but it's inside of the virus now. And then the virus can infect the bacteria, and it does that by injecting its DNA into the bacteria cell. And then they're able to put that liquid that contains the viruses and the bacteria in a test tube into a centrifuge. And when they centrifuge that one, they discovered that the supernate had no radioactivity. All of the radioactivity was in the pellet at the bottom, and that means that the radioactivity had to be inside of the bacteria cells. So the DNA was actually inside of the bacteria cell, and that's what caused the infection and could have the bacteria start to make new virus parts. So Let's do our questions at the bottom. Number 5 says, when the T2 phage was labeled with radioactive sulfur, which structure was radioactive? And you should have found that sulfur made the protein coat radioactive. And then number 6 says, when the T2 phage was labeled with radioactive phosphorus, which structure was radioactive? So the phosphorus labeled the DNA. And then it says, number 7, which part of the virus contains the genetic material, the DNA or the protein coat? And because the DNA was injected into the cell to make new virus parts, we know that the DNA is the genetic material. The next scientist we're going to talk about is Chargaff, who is doing a bunch of research about DNA as well. He made this discovery in 1950. We know that there are 4 DNA bases. Those bases in DNA contain nitrogen, and their names are adenine, guanine, thymine, and cytosine. And so those bases are important to building the structure of DNA. He discovered that the number of adenines is always equal to the number of thymines, and that the number of guanines is always equal to the number of cytosines. So he discovered this ratio where there's always the same number of A's and T's and always the same number of C's and G's. So if, for example, in a DNA strand you have 10 adenines, then you're also going to have 10 thymines. And if you have 7 guanines, you'll always have 7 cytosines. So those numbers are always equivalent, and that's because DNA has 2 halves that are complementary. And the reason that this was helpful for getting scientists to learn more about the structure of DNA is that they were able to figure out that those 2 things were always paired with one another, which we find out later are called base pairs. Next, we'll talk about Franklin and Rosalind Franklin and Maurice Wilkins. So Franklin was the primary source of this information, but Wilkins did share it with Watson and Crick. They were working in the early 1950s, and Franklin made this discovery in 1952. And she used X-ray diffraction to learn about the structure of DNA. So she took crystallized DNA and took an X-ray image of it. And you can see here, this is the picture that she took. And it helped determine what type of structure the DNA molecule could possibly have. Number 2 says, what did this image tell her about the DNA structure? And so she took this picture as though you were looking down a ladder from the top to the bottom, and that ladder was twisted. So you can see in this structure that there's these various little lines that kind of make an X, and that allowed her to determine that the structure of DNA was most likely a double helix. And finally, Watson and Crick are the 2 scientists who discovered definitively the structure of DNA, and they received information that Rosalind Franklin had discovered, and it was given to them by Maurice Wilkins. So they were able to determine the shape of the molecule from Franklin's X-ray diffraction image, and then the base pairing rule that Chargaff discovered also helped them know which molecules were connected to each other in the double helix. So finally, after many years of research with contributions from many different scientists, Watson and Crick, using some of the information that had been collected over the decades, determined that the correct shape of the DNA molecule is, in fact, a double helix. And you can see that picture right here. This is what a double helix looks like. So it looks like a twisted ladder. So if you took a step ladder and then just twisted it so that the rungs crossed each other, kind of like a spiral staircase, you would have a double helix.